So now the important bit is to be able to recognize in pictures which ones are which. So this is your spotter's guide. To neutrophils, eosinophils, basophils, lymphocytes, monocytes, platelets, erythrocytes. These are cartoon drawings once again. So your neutrophils, they're going to be large, clear staining with multilobulated nuclei. Your eosinophils, they're going to be red staining with bilobe, sometimes trilobe nuclei. Your basophils are going to be small, blue staining with bilobe or trilobe nuclei. Your lymphocytes are actually going to be mostly nucleus, very scant cytoplasm, scant meaning almost no cytoplasm. Monocytes are going to be the largest of your white blood cells, excuse me. They're only going to be found in your blood. They are never going to be found in your tissues. And the reason I say never is because once monocytes exit the blood, they are called other things. They become another kind of cell. So a monocyte is never anywhere other than the blood. They will become a different kind of cell once they exit the bloodstream. Platelets are fragmentary cells. They will always be tiny little dots. Erythrocytes are your coin-shaped discs. So here is a peripheral blood smear, so named because it is a smear on a slide of peripheral blood. So right here we can see that we have red blood cells, which are these coins of pink-red stained little discs with that pale center that indicates that you have a smaller, thinner space in the middle and a thicker edge. We have these little blue dots that are these fragmentary cells of these platelets. This guy, I'm going to go out on a limb here and say you can guess what that is. You'll find out later what that is because you'll see this same picture come up again. Good on you if you know what it is already. Shame on you if you skipped ahead to find out. Now, red blood cells, their structure, they contain hemoglobin. That hemoglobin's job is to transport oxygen from the lungs to all parts of the body. They do not have a nucleus. That will be so that they will carry more hemoglobin, leading to an increased transport of oxygen. That biconcave circular shape gives them 30% more surface area for the same volume as if they were just a coin, compared to just being a coin. So that increased surface area to volume ratio is going to increase that rate of exchange of oxygen by simple diffusion across the plasma membrane. Now, a quick reminder about the hemoglobin molecule. You're going to keep seeing that hemoglobin molecule over and over and over and over and over again. The hemoglobin molecule consists of 2 beta chains and 2 alpha chains with 4 iron-containing heme prosthetic groups. All right, and these are the iron-carrying bits. They carry the oxygen. And platelets are an important component of the clotting process. Cambridge didn't ask me to teach you too much about thrombocytes, but I would like you to go to this link. I understand that this link doesn't really go anywhere if you try and click on the screen, except pause the video, and you probably tried to click on it, didn't you? Yeah. All right. Anyway, I'm not being paid to advertise this particular YouTuber, but I am a fan. All right. Now, granulocytes, neutrophils. This multi-lobed nucleus right there indicates that it is a neutrophil. Also the fact that there are no granules that stain. Eosinophil. We have eosinophilic staining granules with a bilobe nucleus. We have here a basophil. I see 1, 2, 3 lobes and blue staining granules. So that makes this a basophil. Now, this is an H and E stain. Hematoxylin and eosin. Every single blue dot on this slide is a neutrophil, unless it also has a little bit of pink right next to it. Yes, I know, you can't focus on it. That's not just the fact that this is probably not 1080p, but whatever. Well, it might be 1080p, I don't know. All right, but eosinophils, you have to get up real close, and even then, some of those aren't eosinophils. So, eosinophils are fairly rare. Now let's talk about the agranulocytes. That right there is a lymphocyte. it is mostly nucleus. There is that tiny little bit of cytoplasm right there. More on that in Unit 5. This monocyte right here with the U-shaped nucleus and huge cell size, this is basically only going to function in engulfing, invading pathogens. But they have to leave the blood in order to do that. So monocytes don't really have that much function in the blood itself. But once they leave the blood, they're no longer monocytes. So a monocyte will become a macrophage. A macrophage down here, this cell all along this border right here, or a dendritic cell. Now, hearkening back to like the early 2000s. Now, this is an H&E stained section of some lymphocytes. But these are a special kind of lymphocytes called plasma cells. We'll also talk about those in Unit 5. We can discuss plasma cells in that these guys have quite a bit more cytoplasm than the lymphocytes that we saw in the blood. And they also have these nuclei that have quite a few of these nucleoli within them. And remember the function of nucleoli to produce the parts that create ribosomes. So they're going to produce a lot of ribosomes, and they have a little bit of cytoplasm in there. And within that cytoplasm, there's a lot of endoplasmic reticulum, rough endoplasmic reticulum. So they've got a lot of ribosomes, and they've got a lot of rough endoplasmic reticulum. The jobs of those two organelles is to produce proteins. So we can surmise, based entirely on those two facts alone, just from looking at these cells, that they are going to be pumping out a lot of proteins. Knowing nothing else, we know that much. When we talk about Unit 5, we will discuss the role of plasma cells as a means of discussing, and as a means of, as a cell that produces plasma, as a means of cell that produces plasma antibodies. Now, blood itself is actually comprised of three different things. Cells, which includes erythrocytes, white blood cells, and thrombocytes, which we have just finished discussing. Plasma itself, which is up to 95% water by volume, but it's usually around 90% if we want to talk most of the time. 6 to 8% dissolved proteins, mostly serum albumins, globulins, and fibrinogens. Some glucose, a bunch of clotting factors, quite a few electrolytes, including sodium, calcium, magnesium, bicarbonate, chloride, etc. Several hormones, quite a bit of carbon dioxide. And then the third component is antibodies. Antibodies plus plasma is called serum. So when you have serum and cells, that's what gets you blood. When you remove the cells from the when you remove the cells from the blood, you have serum. When you remove the antibodies and the cells, you just have plasma. So when you have people who are donating plasma, you have to remove antibodies and you have to remove cells. Or you have to find people who are compatible plasma donors so that you can just take their plasma and just remove the cells so that whatever antibodies are remaining behind, you don't have a bad reaction to. Now, that way they can just be straight serum donors. Now, here we have a capillary bed. And we have our arteriole moving here. We have our capillaries here. And we have our lymphatics here. This is going to be hearkening us back to our vascular system. So that was a brief diversion into blood for a moment. And we're going to come back into our blood flow. Now, remember for a moment about how substances move by diffusion from areas of high concentration to areas of low concentration, passively. So we have high pressure to low pressure, moving blood from this area to this area. That's high pressure to low pressure. We have oxygen moving from high partial pressure to low partial pressure, from arterial to tissue. We have carbon dioxide moving from high partial pressure in the tissues to low partial pressure in the blood. We have glucose moving from high concentration in the blood to low concentration in the tissues. We have amino acids moving from high concentration in the blood to low concentration in the tissues. We have water moving from high water potential in the plasma to low water potential in the tissues, and vice versa. And then from high water potential in the tissues to low water potential in the lymphatic as well. So we have all of these pressure systems and all of these water potential systems and all of these protein systems coming into play here. Remember all of that and review all of that. Go back and rewatch all those videos on how that stuff works if you need to. Remember, high to low, and water follows salt. Now,
cut?
Yeah, we're gonna have to cut.
Okay.